In the second video lecture for Chapter 7, we're going to look at the events that generate action potentials. We also call them nerve impulses. And those are the ways that we can propagate a message down the entire neuron. And we're also going to look at the major regions of the brains and describe their general functions. And so for the first property of neurons is that they're able to respond to a stimuli, and that's what we give the term irritability. So ability to respond to stimuli. Another property of neurons is that they're able to conduct a message from that cell body all the way down an axon. And so by doing that, it can transmit an impulse. And this nerve impulse occurs within 4 steps. Okay. And so I'm going to just give you a brief overview of what this nerve impulse entails. And so really it's a series of reactions. Here is our neuron, here is our cell body, and then here's our axon, and then our axon terminal. Here we have these extensions, and what are these called again? Good dendrites, and they receive all the incoming messages from other neurons or other receptors. Now, if that stimulus is strong enough, we're going to bring that stimulus here within our cell body, and then we can allow this message to carry on from this cell body down this entire axon towards our axon terminals. And so the ability for this to transmit this message, that's what we give the term action potential, or also nerve impulse. Now, at the very bottom of the slide, you see this YouTube link, so that's what you can watch if you want to know more about this nerve impulse, how we can generate it, and what this action potential is. Also in this video, it talks about myelin sheaths, and so it really brings home and gives us understanding of why we even need myelin sheaths in the very first place. And so let's just go over a really quick, brief overview of how this is going to work. And so at rest, our entire axon, inside we're going to have it negatively charged, and on the outside, we're going to have it positively charged. And then we're going to look to see in a few slides why this is so. Okay. So once we have the signal that's large enough, this region between our cell body, here's our cell body, and our axon, right here, this is called the axon hillock. And this is where we're going to start generating and initiating the action potential. So if it's strong enough, what we have is this localized, what's called depolarization. At rest, our cell membrane is at negative 70 millivolts. That's because our inner membrane is negatively charged. What accounts for that? Well, we have a couple of things. We have a high concentration of sodium outside of the cell. Inside of our cell, we have a high concentration of potassium ions, but we also have large proteins, large charged molecules, which give this overall charge of our membrane a negative charge. Now, if our signal is strong enough, what's going to happen? We're going to have this axon hillock be permeable to some sodium. Sodium's going to come in. When sodium comes in, what charge does it take? It becomes more positive. So even though at rest we have a negative 70 millivolt, as sodium comes in, we're going to make our membrane more positive. Now, if the signal to send this action potential down our axon is strong enough, more sodium ions is going to come in, changing this entire membrane to become even more positive. This entire process to make it from negative to positive is termed depolarization. Okay, so once we hit a certain threshold, we're going to undergo depolarization, and then that's when we can start our action potential, which is going to go across the entire span of our axon. Now, once our membrane becomes positive 30, what happens? Well, now we're going to have what's called repolarization. So this sodium influx is not going to stay in one spot. It's actually going to start going towards our axon terminal. And so this area now is going to become more positive, which you can see over here. And by this positive charge, it's going to also initiate and cause more sodium ions to come in. And this area here is going to become more positive. And over time, it's going to keep on sweeping across towards our axon terminal. Repolarization occurs after this localized depolarization happens and passes. And so with this, once sodium comes in, after a while the membrane now wants to go back to its negative charge, Potassium's going to go out. Potassium is positively charged. As that leaves, this is going to build up the amount of negativity found here within our cell. Remember, you have all those protons and other ions. And because of that, our membrane over time, as potassium leaves, gradually it's going to start becoming more negative, and our membrane can then become more of this negative state. Now, over time, what's going to happen? Well, we actually take out more potassium than we need to, and so we go under this short phase where we're briefly more negative than we started, and this is called hyperpolarization. And then over time, what we're going to use is a sodium-potassium pump to take out those sodium ions, bring back those potassium ions inside of our membrane to restore our membrane to a -70 once more. And so that's what happens, starting here from this axon hillock all the way down until we reach our axon terminal. Okay, so let's go through all of those steps that I went through, that I just discussed, and there's 4 of them. So the very first step we're going to go over is this resting neuron and resting potential. And at rest, the plasma membrane is polarized. There are fewer positive ions inside of the cell than outside of the cell. And once again, we have a high amount of sodium outside of the cell, higher amount of potassium inside of our cell, giving rise to this negative charge inside of our cell and positive charge outside of our cell. We also have high amounts of negatively charged proteins. Proteins in itself are actually negatively charged. This is why our membrane is also negatively charged inside. And what helps maintain this gradient is that sodium-potassium pump. So Na plus out and potassium in. Step 2 of this nerve impulse, we have depolarization. And once again, this is going to initiate and get started at that axon hillock. And what happens? A stimulus is going to depolarize the neuron's membrane. Now the membrane becomes permeable to what ion? Good, sodium. Starting off with sodium. And then because of that, sodium channels are going to open. And so the polarized membrane allows all of that sodium to flow inside the membrane. Now, if and only if we have a large stimulus and it's strong enough to allow enough sodium inside of our axon hillock, then we can start this action potential. So the action potential is the movement of ions. So it's when the movement of ions initiates an action potential. I'm just going to abbreviate it as AP. In the neuron due to a stimulus. And then here we have a graded potential or localized depolarization. Localized because we're only dealing with this one area. Will exist where the inside of the membrane is more positive and the outside is less positive. Now what do we have? The next, before our next step, we're going to have this propagation of this action potential. And that's only if enough, what ion again? Sodium enters the cell. And that action potential, we also know it as a nerve impulse, is going to start propagating over the entire axon. So it's going to start going from your cell body towards your axon terminal. Now this impulses will travel faster when the fibers have a myelin sheath. And as you saw in that video that I posted earlier, we have our cell body here, we have our myelin sheaths, and at these nodes of Ranvier, we allow sodium to flood into our cell. Okay, and because of that, we can propagate and move this action potential much faster versus an axon that does not have any myelin in it. We then have repolarization, and so this is when potassium ions are going to rush out. Of the neuron after sodium ions rush in. Which is going to repolarize the membrane. Okay, so we're going to once again make this negative. So repolarization, remember that downward phase of that slope. So repolarization involves restoring the inside of the membrane to a negative charge once again, and the outer surface to a positive charge. So we're returning it back to normal conditions. And then finally, the fourth phase of our nerve impulse is hyperpolarization and refractory period. And so we can restore our initial ionic conditions using what pump again? The sodium-potassium pump. And that's how we can restore back our normal negative 70 millivolt membrane. Okay. Now hyperpolarization, once again, that's going to occur because we have too many potassiums leaving, but over time sodium-potassium pump is going to change that for us and help us return back to our negative 70 millivolts. Okay. So as a review, we don't have this on your slide. I want you to be able to describe the 4 different steps and then also know the sequence of events of that nerve impulse. Okay, so that you will see on your quiz or your next exam for sure. Looking at the next step, what happens when we receive that action potential at an axon terminal? All of this should look familiar to you because this is stuff that we've seen already within Chapter 6. So once that action potential reaches the axon terminal, what happens? Good. Calcium is going to rush in. Oops, so let's get that. Blue. Calcium is going to come rushing in. And what's that going to do? Calcium is going to cause what to occur? Good. These vesicles to fuse with the axon terminal membrane. So it's going to fuse with this plasma membrane. Once that happens, we're going to have the neurotransmitters released into our space between our next neuron. And so here, because we're talking about another neuron, this is a synapse, not synaptic cleft. Synaptic cleft deals with our muscles. And then finally, we're going to have our neurotransmitters bind to our next receptor. Okay, so if it's going to, these neurotransmitters can do a couple of things. If this is a neurotransmitter that tells us to continue to contract and send that message even further down, then we're going to open up those sodium channels. Sodium's going to come rushing in and we're going to lead to another action potential. However, sometimes these neurotransmitters can be inhibitory, in which case we're going to try to stop any nerve impulse downstream. Okay, so only if enough neurotransmitter is released, a graded potential will be generated. And then eventually, an action potential will occur in the neuron beyond the synapse. Okay, so only if we have enough of these neurotransmitters telling us that we want another action potential to occur. Now, looking at all of these neurotransmitters, those electrical changes prompted by those neurotransmitters are brief, meaning that those neurotransmitters are quickly removed from the synapse. So once they bind, they can either be 2 things: degraded, which is a good review, by if we have acetylcholine, what enzyme are we going to break this down with? Acetylcholinesterase, good. And the second way we can get rid of neurotransmitters is how? Good, diffusion out of that cleft. Alright, so here's another video of what events that occur at this neuron synapse. So it's a good thing for you to watch. Okay, looking at the structure of our nerve, we briefly looked at this last class, but we're going to go over more anatomy of the nerves just because we're going to see how these nerves are grouped and how they differ between our somatic and autonomic nervous system. Okay, so nerves, once again, these are bundles of neuron fibers. And they're all bundled by connective tissue. So the first layer we have one neuron. That's encased by this connective tissue called the endoneurium. You take a bunch of these neurons together, you group them up, then we have perineurium, and then this forms a fascicle. So you've seen this terminology before, fascicle. We saw that in our muscular system. Now you take a lot of these fascicles and you put vessels in them. You put them all together and you surround it with an epineurium. And so this eventually is going to form your entire nerve. So for the focus for the next few slides, we're going to look at this division right here. So the motor pathway, looking at the somatic and autonomic nervous system. And that's because we're going to see how we can control these, but depending on how many nerves that we're going to use. And so looking at a nerve in itself, in the somatic nervous system, we're only dealing with one neuron, in which that it originates from the central nervous system and extends directly to the skeletal muscles. Okay, let's give an example. Here is our skin. We have receptors, that's the very first component. We're going to take sensory information to our spinal cord. We can have an interneuron over here, and then directly we have our second neuron that's going to innervate our muscles. And that's how the somatic nervous system, it only deals with just one neuron. Now, autonomic system is going to be different. Here we're going to have our sensory input. We're going to go towards our central nervous system. And then what we have are two neurons. So we have a preganglionic and a postganglionic neuron. And so what does that look like? Well, here, let me change the color so we can see it. We're going to have one neuron stemming here from our spinal cord. Here's our ganglion. Remember, ganglion is cell bodies. Here's our second neuron. Second neuron's going to go towards our muscles. So here's neuron number 1 and then neuron number 2. And here, where we have that cell body, that's called the ganglion. So this is a preganglionic neuron, and this is the postganglionic neuron because it's after. Looking at the effector organs, so somatic nervous system, this dealt only with our skeletal muscles, and so that's why we're doing only their skeletal muscles. Autonomic nervous system, we're looking at both smooth muscle, cardiac muscle, and glands. Somatic nervous system, we have no divisions for that, but autonomic nervous system, we have sympathetic and parasympathetic. Which one again was our rest and digest division? Good, so that's our parasympathetic, rest and digest. And our sympathetic we know as fight and flight. Good. Neurotransmitters. Okay, so from here, if we look at our neurons stemming from our spinal cord going to our skeletal muscles, we're going to release acetylcholine. That's going to cause our muscles to contract. So we know that pathway very well. Here, autonomic nervous system, we can have acetylcholine, epinephrine, or norepinephrine as our neurotransmitters. And so from here, we're going to have our first neuron. It's going to send a stimulus here. We can release either one of these neurotransmitters to our second neuron, and that can send another set of neurotransmitters down here. Now let's take a look at this example when we look at the different divisions. So better illustrations, of course, I'm not a great drawer. Here we have our cell body in our central nervous system. Okay, so this is not stemming from this is not an interneuron. It's the cell body that's found within that spinal cord, and its axon is going to extend into the periphery. And so here it's going to directly release acetylcholine to our skeletal muscles. Once again, we only have one neuron because it's the somatic nervous system. Autonomic nervous system, once again, these are always going to be composed of two neurons. So here we have acetylcholine from the first neuron. It's going to get released as a neurotransmitter. Here we have the ganglion because we have the cell body. And then you have the second neuron over here. It's going to release norepinephrine. Here we have acetylcholine. It's going to get released by the first neuron. And then we have adrenal medulla. So this is a gland. The second gland is going to release epinephrine or norepinephrine into our blood vessels. Okay, so remember it can be two neurons, but here we have in this situation one neuron and then one gland. Second example here, parasympathetic nervous system. We have one neuron. It's going to release its neurotransmitters. Here we have acetylcholine. Second cell body of the second neuron, and that's because here we form this ganglion. And it's going to secrete its neurotransmitters downstream. And once again, we're going to regulate smooth muscles, so digestive system, or cardiac muscle cells. Okay, so we're going to do a little review of this parasympathetic nervous system. And we know this as a housekeeping activities because it conserves energy. It also maintains the daily necessary body functions. And we can remember this as the D division because it's for digestion, defecation, and diuresis, so urination. Looking at the anatomy of the parasympathetic nervous system, we're always going to see preganglionic neurons originating from the craniosacral region. So here's craniosacral region, cranial, and then here's the sacral. So cranial nerves 3, 7, 9, and 10 for here, and your spinal nerves S2 through S4. And so if we look at a terminal ganglion, those are the effector organs. So here's the terminal one. These are going to innervate the organs. And here we're going to have a longer preganglionic neuron. So long preganglionic neuron and a short postganglionic neuron. Neurotransmitter that we're dealing with is acetylcholine. Looking at the sympathetic nervous system, we know this has a fight or flight division. So we're looking at unusual stimuli, and also we can immediately take over our rest and digest system, and we can know this as the E division. And so even though you're resting, you can immediately switch over to this because especially in times of emergency, if you're excited, if you're exercising, or if you're embarrassed. Looking at the anatomy of these two neurons, we're going to have a very short preganglionic neuron followed by a longer postganglionic neuron. And why do you think we really want a short preganglionic neuron? Good, because the moment that we need to activate our muscles and our glands, we need to have that quick communication with our brain and spinal cord and tell us to send that stimulus directly towards the different organs. And so preganglionic neurons, they're going to originate from T1, so thoracic 1, all the way to lumbar 2. And the neurotransmitters we're going to be looking at are norepinephrine, and epinephrine. And so these are going to target our effector organs. Now I also want you to notice that there's of course a lot of overlap. So each of these organs are going to have innervation by both your parasympathetic and your sympathetic nervous system. It's just as we mentioned already, the moment that you need to you're in a state of emergency, even though you're digesting, you can stimulate your eyes, these are going to probably relax, right? The moment that you need to be in an emergency situation, you immediately send that stimulus, that action potential, down your preganglionic neuron immediately, and you can stimulate your eyes to make them more alert. So it's for that quick, fast response. Here's an overview of all the different nerves, preganglionic, postganglionic, and where they all come in and go out. And so I want you to notice one thing. Here we have our spinal nerve. Here we have our ganglion. All of our sensory input is going to come from the dorsal region. Okay, so sensory input is going to come into this dorsal root. And once again, that's that afferent pathway. Okay, and then efferent is always going to come in through this anterior horn or the anterior region of your spinal cord. So this is going to form that ventral root. And of course, it's going to be your motor output. Okay. Now we're going to move on to the next topic, and that's going to look at the regions of the brain. And if we look at the regions of the brain, there's 4 major regions we can separate them into. Majority of our brain, and the mass of that, is going to be our cerebral hemispheres, which we also call the cerebrum. Diencephalon is found right here in the middle. Then we have the brain stem, right here in green. And then finally we have the cerebellum, all the way to the back. Let's start out with the first region being the cerebrum, and here we see a lateral view of the cerebrum, but you'll notice that the cerebrum is actually identical and split into 2 equal right and left portions. And moving forward a little bit, I'll just show you this division right over here. This is called the longitudinal fissure that divides the entire cerebrum equally into half. So it's separated by the longitudinal fissure. And once again, the cerebral hemisphere, or the cerebrum, includes more than half of the brain's mass. If you look at the surface anatomy of the entire cerebrum, we can see that we have ridges and grooves. So we have the gyrus, which is a ridge, and invaginations into that we have our grooves. These are called sulcus, or sulci for plural. Fissures, these are deep sulci. So a fissure is a deep sulcus. And then here we can also notice that we can break and divide our cerebrum into different regions. We have the frontal lobe. We also know that we have that frontal bone in that area. We have the parietal lobe. Also remember we have that parietal bone. Occipital lobe, we have that occipital bone. And then the temporal bone with that the temporal lobe with a temporal bone. And remember, we also have that muscle being that temporalis. So same region, the same terminology. Okay, so we take this cross section. Now we're going a coronal section, or a frontal section of our brain. And we can notice when we cut this in half, we have this long connection between our right and left hemispheres. And this is called the corpus callosum. So this allows communication between both hemispheres. Looking at the layers of the cerebrum, we already saw this before. So we saw the gray matter, and inside we have the white matter. Gray matter being composed of what again? Cell bodies. And the white matter being all that myelinated axons. This slide is just to show you the different lobes of the cerebrum once more, being the parietal, the frontal, the temporal, and then occipital, and that we have fissures dividing the cerebrum into these different lobes. Now looking at the regions of the brain and the cerebrum, we're going to focus on three major areas, and that's going to be the primary somatic sensory area, found here within the parietal lobe. And what this is used for, this is for sending, receiving impulses from the body's sensory receptors. Anterior to that, we have the primary motor area, and this is actually, both of these areas are separated by what's called the central sulcus. And so primary motor area is located in the frontal lobe, and it sends impulses to your skeletal muscles. Another interesting area we have of the cerebrum is called Broca's area, and this is involved in our ability to speak. And someone who has damage to the Broca's area, it's interesting because they can understand you, but they won't be able to vocalize or voice out a response to you. Here's another illustration of our brain, and if we look at it, here's the posterior view, the anterior region. We have the motor in that frontal lobe, and then the sensory in that parietal lobe. And what if you can see this surface of the entire cerebrum, it kind of looks like a map. And really this is a map that it correlates to all the neurons that are going to correspond to these different regions of the body. And so different regions that have more receptors for them are going to have more of an influence and more perception here within your brain. For example, if you want to look at your hand, compare your hand, you're going to have a lot more sensory receptors here compared to something like your leg. And that being the case, I want you to take this as an experiment. Take two pencils, put them at the same width, 1, 2, 3, 4, 5. Okay, or same length, a distance between the two of them. And if you poke those two pencils on your hand, you'll be able to discriminate between the two different points. So here and here. Now if you take that same distance and you put it on the back of your leg, for example your calf, you're not going to be able to feel it as two distinct points. You're going to feel it as one point. And why is that? And that correlates to this example I just told you. So that means that the more your ability to discriminate at smaller distances is the fact that we have more receptors for these different regions of the body. So here we have more receptors for pain, pressure, touch, temperature within your hand compared to somewhere like your hip. Now looking at your motor output, so same thing, you take all the sensory input and you have more receptors for your motor output. Okay, looking at other special senses, olfactory, this deals with smell. This is going to be found within our temporal lobe. Auditory, that's also found in our temporal lobe. Visual, this is found in the back area here, the occipital lobe. Gustatory, so taste. Taste is also going to be found over here, so this is going to be found within our parietal lobe. We're going to move on to the second region of the brain, being the diencephalon, and this sits right above the brainstem. Here that is, and here's our brainstem in green. It's enclosed within our cerebral hemisphere, so it lies deep to our cerebral hemispheres, and it's composed of three parts. We have the thalamus, which is found within the more intermental region. We have the hypothalamus, found more anteriorly, and then we have the epithalamus. And so here is the thalamus. It's enclosed by, this is what's called the third ventricle. Hypothalamus anterior, and then the pineal gland, this is one structure that's part of the epithalamus back here. Let's go over the functions of these different regions. So the thalamus, this is going to surround that third ventricle, and it acts as a relay station for your sensory impulses. Meaning that, let's just scoot to the next picture really quick. Here you can have your visual impulses, so for your visual or your vision sense. And then you have auditory, you have taste, hearing, and smell. All of that sensory input is going to get brought over here towards your thalamus. And then here, thalamus being that relay station for sensory impulses, it can direct all that sensory input to different regions of the brain. So if it deals with vision, where's it going to go? To the occipital lobe. If it's going to go towards smell or taste, we're going to go either to our temporal lobe or our parietal lobe. And so it's really going to transfer all of those impulses to the correct part of the cortex, so we can look to see where it's going on and also interpret our information. Next region we have is the hypothalamus, hypo meaning below, and so it lies right underneath the thalamus. It's an important autonomic nervous system center. And autonomic, these are things that we cannot control, so part of the nervous system we can't have any control over. And so 3 important things it does is help regulate body temperature, in which case we can cause our blood vessels to constrict or relax, and that can also help regulate the amount of heat and dilation within our skin. It can control water balance, and then also it can regulate metabolism. And then finally, 2 other things. It actually houses a limbic system for emotions. So our emotional roller coaster, I guess you can say, is attributed to our hypothalamus. And then finally, it regulates nearby pituitary glands. And we're going to see in chapter 9 which these glands are. So we actually have 2 pituitary glands, anterior and posterior. And hypothalamus, since we're talking about the endocrine system, this in itself will produce 2 hormones on its own. Oxytocin and antidiuretic hormone. And this antidiuretic hormone, which we call ADH, this is really responsible for allowing us to control whether we're going to retain water or we're not going to retain our water. Oxytocin, we're going to find this is used for uteral contractions and for childbirth. So we'll get into that next a couple chapters from now. The next region of the diencephalon we're going to look at is the epithylamus. This forms the roof of that 3rd ventricle, and it houses the pineal body, or we also know it as the pineal gland. And what this is important for is our wake-sleep cycle. Also, what's found in this epithylamus is this pink structure over here, and this is called the choroid plexus. And we're going to find out why this is so important. It's because it forms cerebral spinal fluid. So I'm going to abbreviate that as CSF. This last region that we're going to look at is the brainstem, and it attaches to the spinal cord right down here. And each of the spinal cord is going to run through each of our vertebrae. And so we have 3 parts to their brainstem. We have the midbrain, the pons, and the medulla oblongata. Oops. Looking over here, here is our midbrain, pons, and then our medulla oblongata. And of course, each of these have their specified functions. Midbrain is composed mostly of tracts and nerve fibers, and it's a reflex center for vision and hearing. Looking then at the pons, this is that bulging center that's part of that brain stem, composed of fiber tracts, and it has nuclei involved in the control of breathing. And then the next region of the brain stem that's also controls breathing is the medulla oblongata. So it's found in the lower region of the brain stem. And so we have breathing as well. But we also have heart rate control, blood pressure regulation, swallowing, and vomiting. Now you may be wondering what's the difference between breathing in the medulla oblongata and then breathing in the pons. Well, breathing in the medulla oblongata, this is kind of like the pacemaker of the respiratory rate. So medulla oblongata sets the pace of breathing. And the pons, what that does, it kind of helps smoothen everything out, each of those inhalations and exhalations. Another region that's part of the brainstem, which we don't really look at the structure for this, but it's found actually stemming that entire brainstem region, is the reticular formation. Now, this is a diffuse mass of gray matter along the brainstem, and it's involved in motor control of visceral organs. Part of this is a reticular activating system, and this is also plays a role in that wake-sleep cycle and consciousness. And so here is that reticular formation. Final region of the brain we're going to cover is the cerebellum, and this is kind of looks like a mini brain in its own. It's filled with lots of convoluted surfaces, and what the function of this is for involuntary coordination of body movements. Now, a great example of this is in the event when people give you, not people, but specifically police officers, pull you over and think, not you in general, but people, pull someone over in the case that they think someone is drunk, they give them the sobriety test. And really what that's doing, it's testing the cerebellum. Now, alcohol can cross this blood-brain barrier and enter in an effect and impair our brain functions. And so if alcohol impairs our cerebellum, what's it going to do? It's going to impair our coordination. And so they give you that sobriety test, alternating your hands, touching your nose, and walking in a straight line. And the logic is that if somebody has alcohol, they're not going to be able to coordinate their movements. Okay, and that is it for this next part of Chapter 7. I will see you in the third part of Chapter 7.